MAD protein, and a plasmid expressing either the plasmid vector alone, MAD1, or MAD1 clone. Using antibodies to probe for MSN3A and MAD1 in these lanes shows that the cells express MSN3A regardless of the plasmid they received. The plasmid transfections of MAD1 and MAD1 pro were successful, and now the MAD1 antibody binds to MAD1 pro. Overall, the cell lysate lanes show that MAD1, MAD1 pro, and MSN3A are all expressed where we want them to be. Now we want to look at how our proteins associate. One way is to use an immunoprecipitation column, IP for short. First, pour your solution of interest through a column of beads, and the antibodies embedded in the beads bind their epitope from the solution. By comparing IPs for the flag and flag HDAC plasmids, we can easily control for associations between HDAC2 by comparing which proteins precipitate out. These comparisons are also how we know flag HDAC is expressed where we want it to be. By using anti-flag immunoprecipitation and antibody probing, we can narrow down how MAD1, MSN3A, and HDAC2 interact. Basically, the IP only pulls down proteins associated with the target epitope, so using this data, we can show which proteins interact with which others. In lanes 1 to 3, we IP from cells that have a plasmid containing only flag and a plasmid containing either no MAD1, MAD1, or MAD1 pro. IP with an anti-flag antibody should pull out any HDAC-associated proteins. Probing with anti-MSN3A and anti-MAD1 shows no visible complexes. So since we know from lanes 7 through 9 that MSN3A and MAD1 are expressed, we can conclude that neither of them will bind to the affinity column or the flag epitope. For lanes 4 to 6, let's change it up and use a flag HDAC2 plasmid instead of just flag. And again, one of either no MAD1, MAD1, or MAD1 pro. IP with anti-flag to pull down any proteins associated with the flag HDAC2 fusion protein. You can see via an anti-MSN3A probe that MSN3A precipitates in each of these lanes now that we've added HDAC2, meaning HDAC2 and MSN3A are associated in some way. Let's probe for MAD1. When we don't express MAD1, we don't see it immunoprecipitate, which is what we expect, and no band appears as shown in lane 4. If we express a wild-type MAD1, it does immunoprecipitate, and we see this in lane 5 when the band appears. In contrast, when they express the mutant called MAD1 pro, which we are told does not interact with SYN3A, it does not precipitate. This is shown in lane 6, which does not have a band. Remember that MAD1 pro's mutation removes its ability to bind to MSN3A? Since MAD1 pro doesn't IP out, MAD1's ability to bind MSN3A is necessary for it to come out in a complex with HDAC2. That implies MAD1 binds to MSN3A, which binds to HDAC2. To remind you, we know from the cell lysates shown in lanes 7 through 9 that MAD1 and MAD1 pro were definitely expressed by their plasmids. So let's review the overall implications of this figure. Because SYN3A immunoprecipitates when flag HDAC2 is expressed, but not when flag by itself is expressed. We know that SYN3A binds directly or indirectly to HDAC2. We can see this by comparing lanes 1 to 3 and lanes 4 to 6. MAD1 also immunoprecipitates when flag HDAC2 is expressed, but not when flag is expressed by itself. So we know that MAD1 also binds directly or indirectly to HDAC2. However, we have MAD1 pro, the mutant we are told cannot bind to SYN3A, and since MAD1 pro does not immunoprecipitate with flag HDAC2, we know it does not bind to flag HDAC2 directly. These experiments overall show that HDAC2, MSN3A, and MAD1 form a ternary complex, with the potential order being MAD1 to MSN3A to HDAC2. Thanks for watching, and go blue.
Hi, my name is Ioni Ishan, and this is a video for the Molecular Biology class, MCDB 427 at the University of Michigan. Today we'll be talking about the logic behind Table 18.2 from Robert Weaver's Molecular Biology book. This is Table 18.2, which we'll be coming back to shortly. The main idea behind Table 18.2 is the three-site model of the ribosome. Here we can see the E site, the B site, and the A site. Now, with previous studies done with uromycin, it was already known about the existence of the A site and the B site. This is uromycin, and it kind of looks like a tRNA. It has this part right here that is an amino acid, and the modified adenine. The adenine is modified at this nitrogen right here by the dimethylation. It is often said that uromycin most closely resembles tyrosine tRNA. The main difference being that tyrosine tRNA does still have its tRNA part, whereas uromycin has that replaced by OH. Now, Table 18.2 comes from study 3 tRNA binding sites on Escherichia coli ribosomes by Reinberger, Sternbach, and Knott-Niehaus. What the researchers did is that they found evidence for a third site, which they called the E site. E meaning exit. We'll talk about that later. What the researchers did is that they created 3 radioactive tRNA constructs. The first construct used uncharged tRNA. So this tRNA is supposed to be charged with phenylalanine, but it's really like that phenylalanine. So again, this is just an uncharged tRNA which lacks its phenylalanine. The second construct is a phenylalanine tRNA. So this tRNA is just like the previous tRNA, but in our case, it is charged with its amino acid, phenylalanine. The third construct is kind of special because not only had phenylalanine on the tRNA, but it also the phenylalanine was acetylated. So this kind of was behaving like this tRNA had not only one amino acid on it, but an acetyl group which acted as a second amino acid. So the idea behind this construct was that it was supposed to be an analog of a dipeptide, the dipeptide charge in a tRNA. Now, the researchers ran 2 types of scenarios. The first type of scenario included ribosomes, which had a poly-U mRNA included with them. And the reason why they used poly-U is because it was already known, based on studies done a long time ago, that UU codes for phenylalanine. So uracil, 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 triple uracil codes for phenylalanine. The second type just the ribosome. It didn't have any mRNA. And the research strategy? Well, the strategy was to measure the number of tRNA molecules, so, these tRNA molecules that they created bound for 70S ribosome. And I say 70S because they got the ribosome from E. coli. To measure the binding of tRNA to the ribosome, they used a simple filter binding assay. Now, the main idea behind the filter binding assay is that tRNA, unlike other single-stranded RNAs, does not bind to a nitrocellulose filter. So here in yellow, we can see the nitrocellulose filter, and if we have tRNA here in solution and we pour it through the nitrocellulose filter, it would not stick. It will instead flow through and it will be in the filter. Ribosomes, on the other hand, which contain proteins, will stick to the nitrocellulose filter. So if we have ribosomes and we run it through a nitrocellulose filter, they will stick to the filter. Now, lucky for us, if it happens that the tRNAs bind to the ribosome, and because the ribosome binds to the filter, the tRNA will also be bound to the filter for the ribosomes. So, if we have a solution which has tRNAs bound to the ribosomes, and we run the solution through a filtrate, tRNAs will bind to the nitrocellulose filter. And if we take this filter and we put it under a Geiger or a radioactive scintillation counting instrument, we will see that this will light up, which shows that they will be radioactive. Here, again, let me reiterate, the tRNAs we made were radioactive, and that's why we will see radioactivity. So, now that we talked about all of that, we can take a look at some of the researchers' data. The problem is that here, what we see in figure 1 has a lot more information that we're going to cover in this video, so I went ahead and simplified this figure. Okay, so here we are, much better. So, what the researchers were showing here in the y-axis is the number of tRNAs bound for each ribosome. So, for each ribosome, we see how many tRNAs bound. On the x-axis, on the other hand, we can see how much of that acylated phenylalanine tRNA was used. So, the more we go to the right, the more the concentration of acylated phenylalanine tRNA increases. What we can see here is that as the concentration of acylated phenylalanine tRNA increases, the amount of tRNAs bound ribosome also increases. So in this one right here, you can see the amount of tRNA bound ribosome increasing. But then, once we reach a certain value, you can see that the amount of tRNAs bound per ribosome kind of plateaus at around a value of 1. And this led the researchers to conclude that, once you reach a certain value of acylated phenylalanine tRNA, only one acylated phenylalanine tRNA will bind per ribosome. Now that we talked about that, you got sort of a flavor of the data in the research paper, I will go to the table itself. What the researchers saw is that the acylated phenylalanine tRNA, as we said previously and as we saw previously in figure 1, binds only once when we provide OU, right? That's what we saw before. And they found out that it binds either in the B site or the A site. Phenylalanine tRNA, on the other hand, binds twice if we provide, again, if we provide the OU. mRNA binds twice, and that binding can be both in the B site and in the A site at the same time, whereas the acylated phenylalanine tRNA goes only once in either the B site or the A site. And now, drum roll please. When you take uncharged tRNA and you provide poly-U mRNA to the ribosome, that uncharged tRNA binds to the ribosome not once, not twice, but 3 times. And this was pretty fascinating results. And the researchers found out that the tRNA bound to the B site and in the A site. And the reason why these results were fascinating is because this was clear evidence for the existence of a third site, which is called the E site. And again, E was for exit. It also found out that if you don't give the poly-U mRNA to the ribosome, uncharged tRNA binds only once, and it's in the B site. Phenylated tRNA doesn't bind at all. And acylated phenylalanine tRNA binds only once, and so like before it binds only once, but this time it's only in the B site. But actually, these last results were not from the researchers themselves. This was previously known, and this was from a previous study. So now that we said all of that, now we can talk about some of the major conclusions that we can draw from this study. The first one, and the most important conclusion, is that there is a new tRNA binding site called the E site. And again, like I previously said 2 other times, it's called E site for exit, as the researchers believe that this site can only bind uncharged tRNA. And they believed that this meant that this site bound the tRNA after it had released its amino acid. And it bound this site before it left the ribosome, so E for exit. Another fascinating result is that only acylated phenylalanine tRNA can bind. And to the researchers, this could possibly show that only one polypeptide chain can bind to the ribosome. And again, it's fascinating that this binding could be either the A site or the B site. And this model sort of agrees with information that only one polypeptide chain is produced from ribosome at a given time, right? It's not that the ribosome holds 2 polypeptide chains at the same time. A third major conclusion was that not all binding sites are the same. And for example, one example we can give is that 2 amino acyl tRNAs, right? So as we saw here, 2 amino acyl tRNAs could bind at the same time, right? In this case right here we saw that. But only one acylated tRNA can bind at the same time. And then if we have this case right here where we have uncharged tRNA, they can bind 3 times and it can bind in a new site called the E site. So this clearly shows that the B site, the E site, and the A site are different. And a final conclusion is that mRNA is needed for both E site and A site binding. So when we looked at these 3 cases right here, when we only had tRNAs but we didn't have mRNA, there was no E site or A site binding. So it seems like mRNA is helping the ribosome create those E and A sites, or it's crucial for E site and A site binding. And most importantly, out of these conclusions is the fact that we need the mRNA for A site binding because, as we know now, A site binding is the site where the amino acid tRNA first enters the ribosome. And so this site is important for tRNA recognition. So if the ribosome doesn't have the mRNA, of course it's not sort of unexpected, now that we know all of this, that once we don't have mRNA, we won't have A site binding because there will be nothing there to recognize the incoming tRNAs. That is it, folks. Thanks for watching, and go blue! Hello, my name is Colin Martin, and I would like to welcome you to another video for MCDD 427, which is Molecular Biology at the University of Michigan. Over the next few minutes, we're going to be discussing figures 13.15 through 13.17 from the book Molecular Biology, which is written by Robert F. Weaver. And these figures are going to help us get a better understanding of nucleosomes, their function in preventing transcription, and ultimately the main topic for today, nucleosome-free zones, how they can be located, and how they affect transcription. Now, I want to take just a brief moment to review some basics that you might have already covered in another intro bio class regarding the structure of chromatin. And each of these individual units that we see here are what we call nucleosomes. Each nucleosome is composed of DNA that, when wrapped around proteins called histones, which are colored in blue in this figure, make the DNA mostly inaccessible to RNA polymerase, which inactivates transcription. Now, in order for the DNA to be transcribed, RNA polymerase needs to have a way of gaining access to the promoter of the sequence of interest, and that can only happen when nucleosomes are not associated with that particular region. So essentially, in regard to nucleosome position in chromatin, there are two basic scenarios in which transcription can be initiated. And the first is when a nucleosome happens to be covering the promoter region on a sequence of DNA, which blocks transcription from occurring as it would in this case. A number of chromatin remodeling factors can move these nucleosomes out of the way, however, which would then allow RNA polymerase to activate transcription, which is initiated here as an arrow at the +1 site. The second scenario involves a sequence of DNA whose promoter region is not covered by a nucleosome. And in this case, an activator and any other associated factors can bind the DNA directly and recruit RNA polymerase to immediately begin transcribing the gene. Now, a man named Moshe Yaniv wanted to investigate these relationships further, specifically with regions called nucleosome-free zones. He and his colleagues wanted to determine what kind of evidence they could gather about their existence in the control regions of genes that were able to be transcribed. So they asked themselves, is it true that genes whose control regions are not complex with nucleosomes are transcribed? Are there nucleosome-free regions that we can detect? Well, the first step towards figuring those things out was locating where exactly these nucleosome-free zones were on the sequence of DNA. So Yaniv chose to conduct his experiments using the very well-known SV40 viral DNA. And there are a number of reasons why Yaniv chose to use this specific DNA. And its relatively small size and the depth of knowledge that had been published about its structure were extremely helpful. But another important reason it was chosen was because once SV40 is infected into mammalian cells, and in fact it was monkey kidney cells in this experiment, the DNA forms complexes of nucleosomes that are very similar to our own cellular chromatin. This is where the name mini chromosome comes from, and you'll hear me refer to them as such throughout the video. So one of the first steps Yaniv and his colleagues took was to visualize the SV40 DNA directly using electron microscopy to determine if any nucleosome-free zones existed in its structure at all, and if they did, their position along the strand. And these are the images that resulted. Now, just to orient you, what we're looking at here are two populations of SV40 mini chromosomes, the first being in panel A, and a second population in panels B through D. Each of the mini chromosomes is a circular strand of DNA with a length of 5,224 nucleotides, and each of them has about 24 nucleosomes complexed within its structure. You'll notice here in part A of this figure that these particular mini chromosomes have a very consistent structure throughout, and I've drawn a cartoon version of what we're looking at to give you some other form of reference. Each of the nucleosomes, which are these bigger dots here, is spaced out relatively evenly, so over the 5,224 nucleotide sequence, we're expecting to see a nucleosome every 217 base pairs. These four figures, on the other hand, look a little different, and each of these mini chromosomes, which are still the SV40 mini chromosomes Yaniv was investigating, have a stretch that doesn't have any nucleosomes at all, despite having no difference in the total number of nucleosomes present. And this happened to be occurring in approximately 15 to 20% of all SV40 mini chromosomes that were infected into the monkey kidney cells. Again, you'll notice on this side of the mini chromosome here that there are a significant number of nucleosomes, but there's a stretch on the opposite side of, on average, 248 base pairs that looks totally bare, and this is the nucleosome-free zone. Now, one of the other big reasons Yaniv decided to use the SV40 viral DNA was because previous experiments had shown that treatment of the mini chromosome with a restriction enzyme called BAGO1 makes a cut in the control region for a number of different transcripts that we see here in this image, which I've included to give you a more detailed look at the features of SV40. And we use both and compare exactly what we're looking at. It became of interest to them to see if treatment with the restriction enzymes BAGO1, BAMH1, or EcoR1 would lead to any definitive conclusions about the location of the nucleosome-free zone. If BAGO1 cuts in the control region for the early and late genes in this sequence, and if we could determine that this cut was being made in the nucleosome-free zone, then it's certainly possible that this region, being devoid of nucleosomes, is no accident at all. A nucleosome-free zone in this region could mean all the necessary sequences for transcription, including the promoters, would be available for binding by other transcription factors and RNA polymerase to induce transcription of these genes here that I mentioned earlier. Yaniv and his colleagues needed a way to reliably test these hypotheses, but there was still one issue going forward with the experiments, and that was the fact that SV40 mini chromosome that he was investigating was circular. And to test you a little bit here, I'll pose this question to you. If you were conducting an experiment like this, and you wanted to identify exactly where the nucleosome-free zone was on SV40, why would having circular DNA make your job more challenging? Take a moment to think about it, pause the video, and see if you can come up with a good reason. Well, if you've had a chance to think about it, you may have discerned that without any way of identifying any landmarks, it would be much more challenging to figure out where along this circular mini chromosome you are at a given time. It's important to note that treatment of the mini chromosome with a single restriction enzyme would not be sufficient for determining the location of the nucleosome-free zone. So what Yaniv decided he would do was use high quantities of restriction enzyme to cut the sequence at known positions using BAGO1, BAMH1, or EcoR1. And because Yaniv knew the sequence of SV40, he could draw himself a map of where he believed the nucleosome-free region to be located with respect to these restriction sites. Figure 13.16a on top here shows the locations of each restriction site along the mini chromosome. And part of the genius of this experiment experiment comes from the fact that Yanov chose these specific restriction enzymes to conduct his experiment. Notice that the BAGO1 site is located in the control region of SV40 here on the lower left. Imagine how challenging it might be for RNA polymerase to conduct transcription if there were nucleosomes bound around this region of the DNA. It shouldn't sound like a totally radical idea then that Yanov would propose that of all places along the SV40 chromosome, that if transcription of the genes would ever take place, that this would be the location of the nucleosome-free region. And again, this was just a hypothesis at this point, but by picking a restriction enzyme that would cut near one of the edges of this region, Yanov could experimentally confirm or deny without a doubt whether or not this is where the nucleosome-free region was located using microscopy after treatment of the mutant chromosome with restriction enzyme. Figure 13.16b shows what Yanov expected the products of each restriction cut to be. If BAGO1 cut within the nucleosome-free region as he expected, the result would be a long sequence of DNA bound by nucleosomes with a tail that had no nucleosomes associated with it at all. A cut with either DNH1 or EcoR1, on the other hand, would result in a nucleosome-free region flanked by two regions of DNA bound by nucleosomes. And, as it turns out, this is exactly what Yanov and his colleagues observed. Figure 13.17 is direct evidence of the fact that cutting with BAMH1, which is shown in these first three images, results in the nucleosome-free region located in between two nucleosome-rich regions. Cutting with BAGO1, on the other hand, shown in the final three images here, shows us exactly what we expected. A long stretch of DNA bound by nucleosomes with a tail that is nucleosome-free. I hope that you found this video helpful. I wish you the best of luck in your studies, and as always, go blue.
Hello guys, and welcome to this video, which is made for MCDB 427, that is molecular biology at the University of Michigan. I'm Camille Ayun, and today I'm going to talk about enhancers in general and experiments that highlight their identification and characteristics. So let's start with a brief description of enhancers. What are they, and what do they particularly do? Enhancers are short stretches of DNA, about 50 to 1500 base pairs, that can be bound by proteins called activators to increase the likelihood that a transcription of a particular gene will occur. In this video, as previously said at the beginning, I will start with the discovery of the first enhancer, then I will talk about the fact that enhancers can be downstream of the promoter, and finally I will highlight some of the important features of enhancers, such as it can be moved and it can be flipped. The first enhancer was discovered in the 5' flanking region of evacuating virus 40 gene, SV40. This DNA region has been noticed because of a 72 base pair sequence called the 72 base pair repeat, as you notice here in this figure. So in this figure, you see the structure of the SV40 virus, early control region as discovered, with a starting site right here, with the data box. Here's the starting site, data box, GC boxes, 6 GC boxes, and the 72 base pair repeats. This is just a general description of where the enhancer resided for this particular promoter. Enhancers are frequently found upstream of promoters they control, but this is definitely not a strict rule. Tongawa and colleagues were studying a gene for a heavy chain of mouse antibody called gamma-2b antibody, where the enhancer was speculated to lie within the gene. This figure is a map of the clone gamma-2b gene, where blue boxes are exons and lines between the blue boxes are introns. X2 to X4 are guiding sites for a specific restriction enzyme. Here is the restriction enzyme. So they introduced this gene into mouse plasmacytoma cells that doesn't normally express this particular gene. The suspected enhancer lay in one of the gene's introns as demonstrated by solid line, as mentioned previously, specifically between X2 and X3. The investigators began by deleting two regions of the DNA from this suspected enhancer region. The first deletion here is about 180 base pairs, we're going to refer to this as delta 1, and an overlapping deletion that is bigger than the first one, about 470 base pair, we're going to refer to it as delta 2. Then they assayed the expression of the gamma-2b gene in cells transfected by these mutated DNAs. Keep in mind that they are making these deletions to test the hypothesis that the enhancer was in this region, and if true, then expression of the gamma-2b gene should be inferred. To detect the efficiency of expression of the transfected cells, they incorporated labeled methionine into newly made proteins. Then immunoprecipitated the labeled gamma-2b protein with an antibody directed against gamma-2b. Then they electrophoresed the immunoprecipitated protein and detected them by autoradiography as seen in this figure. For each construct, they are showing 4 independent immunoprecipitation. So starting with lane 1, it is just a control from untransfected plasmacytoma cells. Since no bands immunoprecipitate from these cells, it shows that there is no non-specific binding of plasmacytoma proteins to the affinity column. Lanes 2 to 5 are cells transfected with wild-type gene as reference compared with other constructs. Lanes 6 to 9, transfected gene has the delta 1 deletion. Lanes 10 to 13, transfected gene has the delta 2 deletion. So looking at this figure, there are bands that are related to the heavy chain gamma-2b that we are immunoprecipitating, in addition to bands that are related to the light chain. Antibodies have a heavy and a light chain. So if you are immunoprecipitating the heavy chain, as we are doing right here, you get the light chain too. So comparing the intensity of bands between the wild-type genes and both the delta 1 and the delta 2 deletion, you can notice that delta 1 deletion gave a slight reduction in expression of the gene, but delta 2 deletion gave a profound reduction, which implies that delta 1 deleted part of the enhancer, while delta 2 deleted most, if not all, of the enhancer. So the previous slide was an assay at the protein level. It showed the levels for both transcription and translation. But what if the decreased expression was due to other causes different from enhancers and gene expression? They wanted to measure transcription more directly. That's why the scientists here performed Norton blots with RNA from cells transfected with normal and deleted gamma-2b genes. These blots are shown here in this figure to the right. So lane 1 is a positive control, shows untransfected cells that express the gamma-2b gene. Lane 2 is a negative control with untransfected cells not expressing the gene. Lanes 3 and 4, cells transfected with wild-type gene. Lanes 5 and 6, genes having the delta 1 deletion. Lanes 7 and 8,